everybody. Hopefully you have gone through the background information in Canvas regarding membrane potentials, ion movement, and ion-specific gated channels. If you haven't done that yet, it's best to review that before proceeding with this video. So in this video, we will go over the action potential and how it is propagated down the axon of a neuron. So to understand action potentials, one of the best ways to study it is to view them in their graphical form because it's about the change in membrane potential caused by the movement of ions across the cell membrane. So from now on, when referring to an action potential, or we sometimes shorten that to AP to mean action potential, you'll usually see a graph like this. Okay, so the x-axis displays the change in the membrane potential. So you can see it goes from -70, which is our resting membrane potential, and continues up, gets less negative until it crosses 0 and gets to +30. So again, what that means is the inside of the cell is more negative, starting at -70 compared to the outside. And then if the inside of the cell becomes more positive, then that means you'll see a rise in the graph, okay? And we call that depolarization. When the cell membrane goes back to negative on the inside of the membrane, so going from +30 back down past 0, down -30, -40, -55, right? That's called repolarization, right? And I'll explain the rest of the terms a little bit later, more in context. But also notice on the x-axis, this is indicating time, and this is actually in milliseconds. Okay, so a action potential is very fast, but the timing is also important too. So again, we're going to look at this graph throughout this presentation, and so I'll be explaining the rest of the graph as we get going. So I mentioned that action potentials, or APs, are propagated down the axon of a cell. So here's a reminder of what a neuron looks like. One thing to remember is that a neuron is a cell. So just like any of the cells you learned about in general biology, it's got all the same properties, the cell membrane, the organelles, the nucleus, everything else, but it's just shaped differently. And that's because their main function is communication. So they have to connect to other cells in the body. They're communicating with other cells. So the elongated projection called the axon, that yellow portion that's projecting towards you in that lower picture, that's the axon. And it can be either long or it can be short or anywhere in between. And that's because it might be connecting, starting as a neuron in your brain and connecting to a cell down in your toe. Okay, so sometimes they can be long, or they can be connecting to other cells in your brain or something right adjacent to it. Doesn't always just have to start in the brain. Okay, so again, they can be long and short, but this really demonstrates structure determines function. Remember, we talked about that earlier. So the entire cell is bound by a cell membrane, just like other cells. So cell membrane is just stretching across the axon, the dendrites, the cell body, all that area. Okay. So at the axon, though, the cell membrane is forming a tube. And it's within that tube that has the cytoplasm, and then we'll have all the other things that we talked about inside of a cell. Not so much the organelles, those are mostly up in the cell body, but it also does contain many of the ions. So remember, the inside of a cell, we said, is usually negative at rest. That's because of large proteins that are negative and other anions. Okay, it'll also contain other substances as well, other ions and things. Okay, so it's at this tube where in the membrane where the ions are going to pass through channels. And the membrane of an axon is full of gated channels. So just kind of keep that visualization in your head, and we'll actually see that a little bit later in this presentation. So again, the lower cell is showing you just the axon, that projection coming toward you. The upper cell also is showing the axon, but you can see it's surrounded or wrapped in a myelin sheath. That's that kind of light blue wrapping that you see. So, but more on that later. So we'll get into that probably in part 2 of the lecture, the next video. The other thing to keep in mind is that the action potentials are going to start right where the axon branches off from the cell body. Okay, that region is called the axon hillock. And here's another close-up of that. So again, action potentials propagated from the axon hillock, and you can see it labeled there under the number 2. I'm sorry, it's a little small. But that's the axon hillock. That's the portion of the membrane that gets depolarized first. And if it gets depolarized sufficiently enough, meaning enough electricity stimulates that area enough, then the action potentials are going to run down the axon. Okay, so we're gonna talk about exactly how this happens. Okay, so things to remember about action potentials is that they are very short, brief. So I showed you that graph and it was in milliseconds. We'll see some timing on there later. They're large, so there can be a change of about 100 millivolts in the membrane potential. Okay, so the membrane potential is actually reversing. Remember, the inside of the cell starts out negative and it's going to go toward the positive, and then it'll come back down to negative again. And then when we talk about a membrane action potential, it's actually occurring in a very small segment of the cell membrane. Okay, and what it's going to do is an action potential occurring in one segment of the cell membrane, it's going to stimulate the adjacent segment right next to it. And again, I'll show you some pictures that indicate this. It's all or nothing. That means once an action potential starts, it's going to continue to completion. Okay, it's not going to fizzle out or anything. It's going to continue to completion. If the area of the cell membrane doesn't receive enough stimulation to reach action potential, then it won't run at all. So that's what all or nothing means. Conduction without decrement means that once an action potential is stimulated, that means it will stimulate an action potential adjacent to it, which will stimulate an action potential adjacent to that, which will stimulate an action potential adjacent to that, all the way down the axon. You can see that'll be movement of electricity all the way down. So conduction, okay, it conducts down the axon without decrement. Again, it doesn't fizzle out. It's going to continue to move and stimulate the next and the next. It's not like going to start and then run out of energy and then just stop. That doesn't happen. Okay, so let's look at the graph again. We've added some labels to it. So it's possible you may have to rewind this section of the video a few times to really understand this graph. It's very important that you understand everything that's indicated on this graph. So again, let's run through the graph and get familiar with it. So now you can see across the bottom, it's time in milliseconds. So that just tells you action potentials are very fast. On the y-axis, you see the membrane potential in millivolts. So we measure electricity and voltage, right? And so this is a low voltage within our body, so it's measured in millivolts. Okay? This is actually electricity that we're generating in our body. So don't worry about all the words you see yet. Let me walk you through the graph. So kind of follow what I'm saying. So what we're going to do is we're going to start on the left side of the graph down at the black line, right where it says -70. So -70 is our resting membrane potential. And so you can see the black line even over to the right, right at -70, that line is the resting potential. So again, that means that's the point in a neuron where the movement of potassium ions, and they're moving in and out of the cell because of the leak channels. So remember, there are potassium leak channels. That's allowing potassium to move out based on its concentration gradient, but they're attracted back inside because the inside of the cell is negative and the potassium itself, an ion, is positive. So it's attracted to move back in based on its electric gradient, or we say electrostatic gradient. You can just say electric gradient if you want. But now notice, follow that black line from -70. You see it's starting to move up. Okay, and it moves up to that point at that dotted green line. That's called threshold potential. So what that means is something happened inside of the cell that it became more positive. And that little blue P with the Na plus tells you what that is. And you see the little arrow that's pointing upward. That means that's an increase in the concentration of sodium ions. So sodium ions entered the cell. Okay, and again, that's going to happen at the axon hillock. All of this starts at the axon hillock. Much later in the lecture series, we're going to learn about how that part happens. Not important at the moment. Just know sodium is entering the cell, causing it to depolarize. So depolarization, or before I say that, it is depolarizing, but we'll say it's just increasing to threshold potential. Okay, threshold potential for a neuron is at -50, about -55, -50. Okay, you'll see it a little different in different graphs. Either one is okay. When the axon hillock reaches -50, that means the action potential is going to run. And something triggers it to run. What triggers it to run is the opening of voltage-gated sodium channels. So voltage-gated means it's a sodium channel, it's specific to sodium, that is normally closed, but when the membrane reaches -50, that voltage triggers the gate to open, the channel to open. And when it does, sodium then flows into the cell based on its concentration gradient. So there's more sodium outside of the cell than there is in. It cannot cross a membrane by itself. So once that gate is open, sodium comes into the cell, and that's what the pink writing there means, caused by sodium influx. Influx means it's moving into the cell. Okay, so you can see the cell depolarizes. It means the cell membrane becomes less polarized. A membrane that is polarized means it has a difference in charge from the inside to the outside. So what we're seeing here on these numbers on the y-axis, that's the charge on the inside of the cell. We start at resting membrane potential at -70. The outside of the cell, remember, is positive. So once we hit threshold, a bunch of these sodium channels open, and sodium channels comes flowing into the cell. And as it flows in the cell, that increase in positive ions causes the membrane potential to go to -40, -30, keep going on and on until we even get positive inside of the cell. Now the cell is positive. Until it reaches +30, that gate will close. And that's not because it hits +30, it's just because of the timing. The gate doesn't stay open forever. It only stays open for a certain proportion of time. And that amount of time it stays open, it only allows it to depolarize to +30. And that happens with every action potential. Every action potential has the same strength. It means it only goes up to plus 30. Okay, and so we know that channel closed because when you look at the blue writing at the top, you see that P with the sub Na plus, but now the arrow is pointing down. So that means the concentration of sodium is decreasing because it's not entering the cell anymore. So all of this is what's happening inside of the cell. Okay, it's not entering the cell anymore because the voltage-gated sodium channel closed. But look at right next to it, it says an up arrow with a P and a K plus. That means an increase in potassium ions. Okay, so a new channel is opening. A voltage-gated potassium channel opens, and potassium now leaves the cell. Okay, so when potassium, that's a positive ion leaving the cell, then the cell is going to return back to negative. Okay, yeah, at this moment there might still be some sodium ions inside of the cell, but this part is not about the type of ion, it's just that it's positive. So before positive ions came in, now positive ions are leaving. And that's what the pink writing on the right side there says, caused by potassium efflux. That means potassium is leaving the cell, okay, and it's flowing out of the cell. So we're repolarizing. The inside of the cell is going back to negative. So you see that red line falling downward, and it passes threshold, and it actually overshoots resting potential. Okay, it overshoots resting potential. So let me just define that repolarization. Again, the cell membrane, when it repolarizes, it's becoming more polarized. We're going to move away from the positive like it is outside of the cell, and we're going to go more toward the negative. Okay, so it's repolarizing. But it overshoots the resting membrane potential, and it goes more negative than usual. It goes down to negative 80. That portion is called hyperpolarization. Okay, and then it returns itself to resting potential. Alrighty, so that's just an explanation of the graph. What I want to do now is explain these different types of channels. The voltage-gated sodium channels, the voltage-gated potassium channels, and then I'll also explain how it gets back to resting potential after it hyperpolarizes. All right, so let's start with voltage-gated sodium channels. Let me zoom in on this so you can see it better. So this is basically showing one voltage-gated sodium channel at different moments of time as we go across the graph here, across the picture. So the very first voltage-gated sodium channel that you see is the voltage-gated channel when it's closed, okay? So remember, I keep saying voltage-gated. That's because you see the little gates there. One is closed, one is open. Okay, those gates will shift their position when there's a change in the potential of the membrane right in the area where it's embedded. So when the change in the potential of the membrane occurs, that's the stimulation to open up these gates. And it will change the position of these gates. So you see the one gate that's closed, called the activation gate, it will swing open. And then the other gate, called the inactivation gate, that looks like a ball on a string of pearls, that will swing closed. Okay, the timing of those two gates is important. All right, so voltage-gated sodium channels have a double gate. So whenever you're talking about your channels, you do need to indicate to me, don't just say sodium channel. In this case, we're talking about voltage-gated sodium channels. That tells us that it is electricity that stimulates it to open, okay? Because later on, we're going to talk about a voltage-gated potassium channel. And we have already spoken about leak channels for potassium. That's going to be different, Okay, so it's very important you use your terminology correctly. Okay, so I'll walk you through this picture right now. So let's start with the first one. This shows you that the membrane is at resting potential. And so at the very bottom there, it says resting potential negative 70 millivolts. And in that condition, the activation gate is closed and the inactivation gate is open. The position of those two gates tells us that while the channel is closed and sodium isn't allowed to come in, it can't cross the membrane by itself. It needs an open channel. So while the activation gate is closed, this voltage gated channel has the potential to be stimulated to be opened. Okay, it's ready to go. It's just waiting for a change in that resting potential. Okay, the next picture, the one in the middle, this is just a different moment in time. So it's the same channel, but a different moment in time. And what happened at this moment in time is the membrane reached threshold potential. So at negative 50, that stimulated both gates to change their position. But the activation gate is faster. So the activation gate swings open and it stays open long enough for sodium ions to flow inside, causing the inside of the cell, the intracellular fluid, ICF, to become more positive. Okay, and again, this is about timing. It only stays open, so it stays open, but the inactivation gate is slower. So in the next panel, the third picture there, the next moment of time, now the inactivation gate slowly swings closed and it stops the influx of sodium. And that happens to be at a time when the inside reaches positive 30. Again, it's not because it reaches positive 30. It's just that that's the timing when the inactivation gate finally closes. So I'll say it again. Both gates are stimulated to change their position at the same time when in that middle column, the threshold potential is reached at negative 50. But the activation gate changes its position quickly and it opens. Sodium comes in. Then the inactivation gate catches up and it closes. It's slower. So once it closes, it inactivates this voltage-gated sodium channel. Sodium can't come in anymore. And while that gate is closed, no amount of change in resting potential will open it again. It's inactivated. But again, it will slowly change its position in time. And we'll get to that part later. Okay? So one thing to remember is that this sequence of events with this voltage-gated sodium channel is happening during the depolarization phase of that graph. Later on, we're going to layer these gates with the graph. So don't worry about it if you're not seeing it yet. Okay, so when the voltage-gated sodium channels are stimulated, the voltage-gated potassium channels are stimulated at the same time, but they're even slower to open than that inactivation gate is to close. Okay, so when they do finally open, then we enter the repolarization phase. So again, let's look how the voltage-gated potassium channels open. One thing to notice here, there's a single gate. You don't have to worry about that double gate. The double gate is going to have some importance a little bit later, okay, about the timing of it. We'll explain that later. Okay, so in this slide, just like the other slide, this is the same voltage-gated potassium channel, but just in different moments in time. So the first one shows you it's closed, and that happens at rest. It gets stimulated to open at threshold at negative 50, but it's so slow to open that it doesn't finally open until the action potential peaks at that positive 30. Again, it's about timing. It's not about the fact that it reached positive 30. It's just the timing. That's where this one happens to open. When it opens, then the graph will start to fall again and will repolarize, because when this opens, remember that potassium likes to move out of the cell. There's more potassium in the cell than there is out. So it tends to move out of the cell when a channel opens. Okay. And remember, right now, the inside of the cell is positive, so it's not moving along. It is actually moving along an electric gradient because the inside of the cell is positive, so it wants to move outside. Okay. Mostly it's dependent on its concentration gradient. So again, this gate opens, potassium is moving out, and so now the cell is repolarizing. The other thing to remember is, when we learned about resting membrane potential, we said there were potassium leak channels. These are potassium channels that are always open. So remember, channels are specific to their type of ion. Only that type of ion can move through it. That's why I'm calling it a potassium leak channel. So now there's even more potassium channels open. We've added the voltage-gated channels. So more potassium is moving out than normally. And so we actually hyperpolarize. The inside of the cell becomes more negative than resting potential. It goes to negative 80. Okay? It will eventually bring itself back to resting potential. And again, I'll explain a little bit later why. So again, what's important here is it is a specific ion moving through, but the fact that it's just positive ions moving out now is causing the inside to become more negative. All right, let's put the opening and closing of each of these voltage-gated channels together with the timing of the action potential graph. Okay, so at the top, what we see is kind of the greenish color is the outside of the cell, the extracellular fluid. Okay, and that's normally positive, and partly because there's a lot more sodium ions outside of the cell than there are inside. The kind of that peachy color indicates the inside of the cell, which is the intracellular fluid, the ICF. And so that is more negative. Okay, and that's because there are more anions inside of the cell, or negatively charged ions, and also proteins that are negatively charged. And the proteins are trapped inside of the cell. They're too big to get out of the cell. Okay, they have a function inside of the cell themselves. There's also more potassium inside of the cell, but that positive ion, the concentration of the negative particles is actually more than the positive ion of potassium, and so therefore, again, the inside of the cell is negative. And at resting membrane potential, you see in the graph below, you see a black line starting at negative 70. Now you see some numbers along the time with milliseconds. So this is only going to take about 6 milliseconds. That's very fast. Okay, so at resting membrane potential, we're at negative 70. That means the inside of the cell is negative 70. Look up at the voltage-gated channels. The pink one is the sodium voltage-gated channel. The activation gate is closed. Okay, and the voltage-gated potassium channel is closed. There's only one gate for that one. Okay. Also to note, on the voltage-gated sodium channel, the inactivation gate is open. So this means it's ready to be stimulated. Okay, it can open at this point. Here we go. So something's happening inside of the cell so it says there on the graph, depolarizing triggering event. And so you can see the graph is rising, And if that depolarizing trigger event hits -50, our action potential is going to run. If it doesn't hit -50, our action potential won't run. Our nerve won't be stimulated. And that, again, is a later explanation of how it reaches -50. So don't worry about that right now. Let's worry about these voltage-gated channels. So at -50, again, the three gates, both of the gates on the sodium channel are stimulated and the gate on the potassium are stimulated. But the activation gate of the sodium channel is fast. So it opens first. When it opens, sodium moves in. And so now you can see that sodium ions will pair with the negative ions, and it's actually causing the inside of the cell to become more positive. The outside of the cell is losing positive ions, so the outside is coming slightly more negative. Okay, but you can see a rise in the graph. So we've just stimulated depolarization. And again, this is all or nothing. So it hit -50, so this action potential is going to run. It's always going to be the same strength, so that sodium voltage-gated channel always stays open. The activation gate stays open long enough so that the action potential will reach +30. Okay. Next slide. And it's still open, and the depolarization is still occurring. We're still rising. We're going past 0, and now we're into the positive. Now the timing, we've reached about, what is that, maybe 2.5 milliseconds or so. That's enough time now for the inactivation gate to finally close. It was stimulated at threshold, but it took this amount of time to finally close, about 0.5 millisecond to close. Whereas the activation gate opened immediately, it took the inactivation gate about 0.5 millisecond to close. So no more sodium ions or any positive ion is entering the cell anymore. Okay, so at the same time, you see our potassium voltage-gated channel is opening. So again, that one also was stimulated at threshold, but it took about 0.5 millisecond for it to finally open. So as it opens, potassium now can move out. Always keep straight your concentration gradients of sodium and potassium. They're in normal resting potential. There's more sodium outside and there's more potassium inside. That means when a gate opens, sodium wants to move in and potassium wants to move out. Also, you should know the direction, which way sodium and potassium moves. It's about their concentration gradients. So again, look at the graph. Our sodium channel opens. Sodium now is flowing out. Positive ions are flowing out. So we are repolarizing. There's a fall in the red line of the graph. We're repolarizing. And at the same time, they're not showing you this part of the membrane, but there's potassium leak channels that are always open. So even more potassium is flowing out than normal. So you can see down at the bottom there, a little purple line, we're hyperpolarizing. We went past threshold. We're going to -80. And that's about the point where our potassium voltage-gated channel is closing. And so now there's no more sodium movement and there's no more potassium movement through these two types of channels. Okay? And, but look, we hyperpolarized. Both of these channels are closed, but we've gotten back, we've increased back to resting potential and we stabilize out. So something must be causing that to happen. Well, that something are sodium-potassium pumps. So this is another type of channel, but it's a pump because it uses ATP. And what it does is it pumps the sodium out and it pumps the potassium back in. And so this next graph or next slide will kind of show you that a little bit better. So this is what's returning the membrane back to resting potential. These are scattered throughout the membrane. They're always running, but once those two voltage-gated sodium channels and the voltage-gated potassium channels close, then these sodium-potassium pumps can catch up. So it's called a pump because it's using energy in order to open. It actually flips. You can see it crosses the membrane. And once so sodium will attach to the inside of it from the inside of the cell because we have extra sodium in there. We want to kick it out of the cell. That's where it started, so we want to put it back. Okay, so 3 sodium ions attach to the inside of the cell or inside of the pump, and ATP attaches to it. Okay, ATP then splits and ADP is released, and that causes that protein to flip open to the outside. And so it causes that sodium to be pumped out. We use the word pump because it is actively using energy to do that. Once it's open to the outside of the cell, the potassium is attracted to it. And so 2 ions of potassium attach to the inside of it. And you can see at the bottom there, the little blue spike, that's the inorganic phosphate that's still attached. It releases, and that causes the pump to flip back to its original position, opening to the inside of the cell, releasing the potassium inside. Okay, so these things are just restoring the cell back to homeostasis. It's pumping the sodium back out, and it's pumping the potassium back in. If it couldn't do that, then the neuron wouldn't be able to generate another action potential. So it's important that these sodium-potassium pumps keep running. So just a few more things to remember about action potentials. So again, when sodium channels open during depolarization, they do that by positive feedback. So let me show you the next slide first so we can stay on that topic. Okay, so this is positive feedback because depolarization is what increases the membrane potential, right? That depolarization opens the voltage-gated sodium channels, which allows sodium in. Sodium depolarizes the membrane, which opens more sodium channels, which causes more sodium to come in, right? So we're getting more of the same. That's a positive feedback loop. Okay, it doesn't stop until those channels are closed. Then we need to restore the cell back to homeostasis, so our sodium-potassium pumps help do that. Okay. Next one, when the sodium channels become inactive, then the channels for potassium open. So it's a sequence of events. That sequence is important. That's going to cause the repolarization. And again, the action potential at one point in the plasma membrane, at one section, that's going to generate an identical action potential right next to it, adjacent to it, which will cause the next action potential to run adjacent to that. And you can see that's movement, that's electricity moving down the neuron. That's how it goes from the cell body down to the axon terminals. Okay, so it travels along the plasma membrane undiminished because it's starting, and that's important, it's starting a new action potential right next to it. So remember, if a new action potential gets triggered, it's going to run at the same strength. And it will trigger a new action potential because we always get it back to -50 at least in the next segment. So that flow of sodium that comes into the cell kind of flows adjacent to it and opens up the next channels. Here is a better picture illustrating that. Okay, so we're at the axon hillock right by the cell body and right where the axon starts. And this actually gives you a little insight to what's causing that. It's something called a graded potential. That's what increases the threshold at the axon hillock. And that's what I'm going to discuss in the next lecture. Okay, so that triggering of threshold causes sodium ions to come in, and now you can see the inside of the membrane is becoming positive. And that first graph shows in that segment of the membrane where it's yellow, that's an action potential running there. Notice though, in the next segment of the membrane where it's kind of a light yellow, sodium ions are starting to flow in that direction, and that area is starting to reach threshold. Boom, and there it is. It reaches threshold, and the next action potential now is triggered while the first action potential is going through its repolarization phase and reaching hyperpolarization. In that middle one now, where that second action potential is running, it's beginning to trigger the third action potential right next to it. Okay, and so on and so forth. That just runs down the membrane. And so you can keep going until it gets to the very ends of the axon. You can see it splits into what are called the axon terminals. It's also known as the synaptic terminal. And that, again, is going to be a next a lecture, maybe two lectures from now, I think. Okay? So we're going to see what happens as action potential does something to that synaptic terminal. It's actually going to release neurotransmitter. So I'm going to end this lecture, and I'm going to continue with the next lecture to explain a little bit why the inactivation gate and the activation gate is important, because one of the things you'll also notice on this picture is that an action potential can only run from the cell body to the synaptic terminals. It can never run backwards. And the reason has something to do with the inactivation gate. All right. So see you guys in a little bit.